all, welcome and thank you for joining us for this webinar. My name is Lindsay Nelson. I am the Life Science Technology Platform Leader for Oncology Reagents at PerkinElmer. Today's webinar is titled Innovative Approaches to Investigating KRAS Therapeutics with Biochemical and Cell-Based Assays. I'm very pleased to introduce our presenter, Elodie Dupuis, an R&D project manager and team leader focused on developing homogeneous assays, including kits specifically for KRAS research. Take it away, Elodie.
Thank you, Lindsay. And hello, everyone. Thank you for joining us today for this presentation on new KRAS research solutions. Today, we are going to look at some innovative biochemical and cell-based assays to study KRAS and SOSङ binders, as well as some research applications they open and the kind of results they achieve. Before we dive in, let me explain a couple of things about the presentation. First, this will be followed by a Q&A session, so please, all your questions until then. Second, this presentation will quote data from different sources, including notes that have been published by PerkinElmer. All of these will be shown at the end of this presentation. First up, let's have an introduction to RAS proteins, what they are, what they do, and why they are of interest to us. More than 150 human RAS proteins have been identified and classified in 6 families. These proteins are important, playing a role of signal transducers for pathways that regulate numerous intracellular functions, including cell growth, proliferation, differentiation, and survival. RAS protein mutations have been found to drive more than 30% of all human cancers across a wide range of cancer types. The KRAS isoform accounts for 85% of all RAS mutations. Thus, intense research initiatives are underway globally to develop effective therapeutic approaches for KRAS-driven cancers. The human genome contains 2 copies of the KRAS gene. KRAS2 gene has 6 exons, and alternating splicing of exon 4 produces 2 mRNA forms, which in turn give rise to 2 protein isoforms, which are KRAS4A and KRAS4B. The KRAS4B variant predominates in human cells and is commonly known as KRAS. At PerkinElmer, we are working with this variant. KRAS gene mutations occur most frequently at codons 12, 13, and 61, and are present in many types of human tumors, including pancreatic, colon, and lung carcinomas. The diversity of KRAS mutations that cause human cancers and the persistent difficulty in identifying new mutations are the drivers for multiple types of KRAS therapies being developed and that are already in use. In terms of background, moving on to KRAS and SOSङ as key interacting protein partners implicated in many cancers. KRAS are G proteins that contain 4 domains, including a conserved GDP-GTP binding domain and 2 switch regions that determine how they react to their substrate and environment. Conformational changes in the switch regions allow KRAS proteins to have many potential configurations and interaction with various partners such as guanine nucleotide exchange factors, the GEF, promoting formation of the KRAS GTP bound active form, the on state. GTPase activating proteins, the GAP, promoting formation of the inactive GDP bound form, the off state, and effectors promoting activation of downstream cascade of signaling pathways. In a normal physiological context, KRAS is mainly in the inactive GDP-bound state. The conversion of KRAS from its inactive form to the active state is mediated by GEF. The GEF of interest is SOS1, which is a multi-domain protein containing both N-terminal and C-terminal regions joined by a helical linker which acts as a hinge, allowing SOS1 to adopt both an active membrane-bound conformation and an auto-inhibited cytosolic conformation. The interaction between GDP-KRAS and the catalytic site of SOS1 leads to GDP release. An allosteric binding site also exists, functions as a positive feedback loop, and binds GTP-KRAS more strongly than GDP-KRAS, promoting activation of SOS1 catalytic activity upon KRAS binding. There are two approaches to targeting KRAS: selective KRAS therapies and pan-KRAS therapies. Selective KRAS approaches target one oncogenic mutant, whereas pan-KRAS therapies target multiple KRAS mutations, and the direct inhibition of SOS1 belongs to this class of therapies. Now, moving on to map kinase I readouts in KRAS oncogenic cellular contexts. Activated KRAS activate effector proteins that promote downstream cascade of signaling pathways and promote cell cycle progression through, say, replication, growth, differentiation, and chemotaxis. KRAS mutations that cause oncogenic activity and different characteristics in different cancer types result in KRAS protein stuck in the GTP-bound form. When a KRAS protein is stuck in the on-state position due to the mutation, downstream effector pathways also remain active and tumor proliferation remains high. Investigations for specific inhibitions of KRAS downstream pathways are of interest in cancer research. Today, we will be discussing new solutions upon traditional approaches focused on KRAS and SOS1 as partners in biochemical and cell-based contexts for KRAS therapeutics research. First, PerkinElmer has developed a portfolio of biochemical assays to characterize KRAS and SOS1 binders as inhibitors for their interaction. PerkinElmer facilitates the screening of KRAS SOS1 inhibitors by providing commercially available, no-wash, and ready-to-use homogeneous KRAS SOS1 binding kits for KRAS SOS1 analysis. Available in both AlphaLISA and HTRF technologies, these kits come with detection reagents, assay buffers, tagged recombinant proteins, and optimized technical procedure to perform the assays with no optimization necessary, and robust validations with compound of reference to bind KRAS wild type, KRAS G12C as mutant, and SOS1. The assay principle goes as follows. Compounds, then recombinant and tagged KRAS GTP and SOS1 proteins are dispensed in miniaturized plates. Finally, HTRF and AlphaLISA detection reagents based on anti-tag strategies are introduced prior to the incubation for 2 hours. In terms of what the assays provide, they are biochemical assays designed to identify specific inhibitors targeting KRAS G12C SOS1 from KRAS wild-type SOS1 complexes. We have good demonstration of that specificity on the slide. In the corresponding experiments, a panel of reference compounds have been tested and the pharmacological ranking obtained was well correlated with the specificity described in the literature. Data are not shown here, but the specificity was both demonstrated with HTRF and AlphaLISA kit versions. Also, The results demonstrated the ability of the kits to identify inhibitors, which can be SOS1 binders, illustrated here with the BAY compound, or KRAS binders, illustrated with all the other compounds shown here. As shown on the slide, the same experiments were also performed with the GDP as competitor nucleotide of the GTP, in order to illustrate the applicability of the KRAS SOS1 binding kits to also identify some compounds that are actually GTP competitors as positive hits by disturbing the binding of GTP on KRAS, which is required for the detection of the interaction with SOS1 in this biochemical context. In order to identify the GTP competitors among the positive hits, additional investigations can be performed with the PerkinElmer's HTRF and AlphaLISA KRAS G12C and wild-type GTP binding kits. PerkinElmer facilitates the screening of KRAS GTP competitor by again providing commercially available, no-wash and ready-to-use homogeneous KRAS binding kits for KRAS G12C and wild-type analysis. Available in both AlphaLISA and HTRF technologies like the KRAS SOS1 kits, these kits come with detection reagents, assay buffers, recombinant proteins, which are also tagged, and optimized assay protocol to perform the assays with no optimization necessary, and robust validations with nucleotides of reference to compete the GTP on KRAS wild-type and KRAS G12C. The assay principle goes as follows. Compounds then tagged recombinant KRAS proteins are dispensed in miniaturized plates. Finally, HTRF or AlphaLISA detection reagents based on anti-tag strategy and fluorescent or biotin GTP derivative are introduced prior to the incubation for 1 or 2 hours. In terms of what the assays provide, they are biochemical assays designed to identify specific compounds competing the GTP bound in KRAS G12C or wild type. We have a good demonstration of that competition on the slide. In the corresponding experiments, a panel of reference nucleotides and reference KRAS SOS1 binders have been tested. The competition profiles obtained with all guanosine nucleotides and the absence of full inhibition detected with all KRAS SOS1 binders were consistent with the data from the literature and the binding sites described for KRAS and SOS1 binders. Data are not shown here, but the competition was both demonstrated with HTRF and AlphaLISA kit versions. Also, the kits enable the fine-tuned characterizations of KRAS GTP allosteric compounds for their binding close to the GTP binding pocket on KRAS. Finally, the KRAS GTP binding kits enable the discrimination of GTP competitors or GTP allosteric modulators among a panel of screened compounds from a chemical library additionally to the KRAS SOS1 binding kits. Now, let's take a look at a summary of what kind of studies all these assays are aimed at. First, the KRAS SOS1 binding kits enable the identification of positive hits as competitors of GTP and binders of KRAS or SOS1 for inhibition of KRAS SOS1 complexes implicating KRAS GTP bound conformation. They enable the discrimination of specific inhibitors of KRAS G12C SOS1 complexes from the KRAS wild-type SOS1 ones. Finally, They enable the pharmacological characterizations of compounds for their potency, efficacy, and ranking. Second, the KRAS GTP binding kits enable the discrimination of GTP competitors and allosteric modulators among the positive hits detected with the KRAS SOS1 binding kits. Third, the KRAS portfolio provides additional tools to further characterize KRAS SOS1 compounds' properties and elucidate the mechanism of interactions between KRAS SOS1 and the GTP in a biochemical context. Altogether, the PerkinElmer's KRAS binding kits provide new biochemical solutions to screen pharmacological compounds and estimate the potential of positive hits to become a therapeutic tool for cell-based investigations. PerkinElmer also provides a comprehensive portfolio of HTRF and AlphaLISA surefire ultra phosphototal cell-based assays that help characterization of KRAS SOS1 inhibitors on downstream KRAS signaling. These cellular signaling assays enable discrimination between the cellular action of the hit compounds and evaluation of their efficacy and potency in modulating the downstream KRAS pathways. These kits represent efficient tools for the estimation of a therapeutic profile for KRAS targeting compounds in various cancer cell lines. The assay principle goes as follows. The assays were designed using a classic immunoassay sandwich format based on the detection with antiphospho and antitotal antibodies specific to the protein as readout to be monitored. To perform the assays, cells are dispensed in microplates, stimulated, then lysed prior to detection. In terms of what these assays provide, they are cell-based assays designed to evaluate in a cellular context the compounds characterized in KRAS SOS1 biochemical assays and discriminate the specific KRAS G12C inhibitors from the wild-type ones. We have good demonstration of that specificity on the slide in a cellular context. In the corresponding experiment, 2 KRAS reference inhibitors were tested on the HT29 cells as colorectal cancer cells in which only KRAS wild type is endogenously expressed. As expected, results demonstrated the inhibition of phospho-ERK and phospho-MEK mediated by the BI inhibitor, which is described in the literature to bind KRAS wild type, instead of no inhibition was detected through AMG stimulation described in the literature to be specific to KRAS G12C. That are not shown here, but this specificity was both demonstrated with HTRF and AlphaLISA surefire ultra-quick versions. Results confirm the specificity of pharmacological compounds previously characterized with KRAS SOS1 biochemical assays. The PerkinElmer's phosphototal cell-based assays portfolio also provides solutions for fine-tuned characterizations of SOS1 binders in a KRAS downstream signaling cellular context. Here, we have a good demonstration of the kind of results provided. In the corresponding experiments, the SOS1 binder BAY was tested on 2 different cancer cellular models. First, the KLU1 cells as lung cancer cells in which only KRAS G12C is endogenously expressed, and second, the Ki562 cells as leukemia cells in which only the KRAS wild-type version is endogenously expressed. Results demonstrated that there is no effect on total ERK and that there is an inhibition of phospho-ERK in both cellular models, but interestingly, the potency and efficacy of the SOSone binder to inhibit phospho-ERK as KRAS downstream signaling readouts were different depending on the cellular model tested. The same conclusions can be made from results obtained both with HTRF and AlphaLISA Surefire UltraKit versions. The conclusion of that study is that by combining all PerkinElmer solutions, including HTRF and AlphaLISA KRAS biochemical assays, and the HTRF and AlphaLISA Surefire Ultra Phosphototal cell-based assays, fine-tuned characterization of KRAS, SOS1, and GTP binders can be performed to correctly identify compounds as inhibitors for new therapeutic strategies in oncology. To conclude this presentation, I wanted to take a look at the current PerkinElmer's catalog for KRAS investigations. The listed HTRF and AlphaLISA KRAS biochemical kit versions are available and can be found online. Regarding the cell-based signaling assays, some of the relevant kits are listed here, but many others are also available and can be found online too. Finally, before closing this presentation, I wanted to highlight the library of content that PerkinElmer has created for KRAS researchers and scientists, including 2 application notes with results and technical info about KRAS assays, a white paper for more educational purpose, and a guide with the key guidelines to running cell signaling experiments. That's all for this presentation. Thank you for your attention, and we will now start taking questions.
Okay, thank you so much, Elodie, for that presentation. At this point, we will take any questions that you may have. As a reminder, on the right-hand side of your screen, there's a little chat box to enter any questions you may have, and we will answer as many as we can as time allows. So it looks like we have a few questions here in the queue. Elodie, the first question is, are these types of assays amenable to miniaturization?
Yeah, so these assays were optimized in 384 plates, and we didn't test any other assay formats. But with these types of assays, and especially for the biochemical assays, with AlphaLISA and HTRF technology, it's totally possible to perform these assays in a miniaturized format to the 1536 well plates. So yes, these assays can be miniaturized by optimizing volumes of reagents which are dispensed in the plate.
Okay, thank you, Elodie. Second question is, why did you choose phospho-ERK as a readout in the cell cancer model?
Good question. So we chose phospho-ERK as a downstream readout in KRAS signaling for several reasons. And first, because ERK is a member of the 2 main signaling pathways related to KRAS. So it's a really relevant readout to characterize KRAS inhibitors in cells. And also because ERK is one of the more described readouts for active KRAS signaling, and it can be found in the literature several papers mentioning KRAS inhibitors which were tested in different cancer cells So we could find any values regarding specificity, potency on various type of cells. And finally, because phospho-ERK is implicated in numerous cancer cellular processes, including, for example, migration, apoptosis, growth, differentiation, and so on. So it's really a relevant readout.
Great. Another question, how could you account for cell toxicity of the compounds for your cell-based assays? What would your recommendation be?
I'm sorry, Lindsay, I didn't hear the question.
Oh, sure. So the question is about how could you account for cell toxicity of the compounds for your cell-based assays? What would your recommendation be in terms of looking at maybe some toxicity of compounds when running cell-based assays?
In fact, you cannot monitor cell toxicity by using only these assays, but it's really useful to compare the phospho-ERK, for example, and the total ERK. And if you don't detect any decrease of signal, it because that there is no cytotoxicity effect. But additionally to this kind of assay, you can also perform other assays, and for example, our ATP light assay.
Great, thanks, Elodie. The next question we have is it correct that the screening with the KRAS SOSone binding kit will also identify GTP inhibitors?
Yes, it is, and because for these KRAS and SOSone biochemical kits, the interaction between both proteins is driven by the concentration of GTP, which is mandatory to detect this interaction, meaning that if a compound competes with the GTP, the interaction between KRAS and SOSone will be altered. So AlphaLISA and HTRF signals will decrease with these kits in the same way as for KRAS or SOS1 binders, which inhibit the interaction.
Thank you, Elodie. Let me just check the queue list. Another question is, do you have assays optimized for other RAS isoforms, such as NRAS or HRAS, and any of their respective mutations?
Not for the moment, but I guess it would be very interesting to develop this kind of assays. So maybe we will have these kits in the future.
Great, thank you, Elodie. Let's see one more. So someone asks that in your slide 13, Elodie, phospho-ERK and phospho-MEK signal does not reach the bottom of the plot. Can you maybe explain the reason for this? Is this normal, or could this be improved?
In fact, we observed that for some compounds, the efficacy to inhibit the phosphorylation of ERK is totally different depending on the cells. So for the compound which is presented on this slide, so it's a BI, we observe that it's only a partial inhibition of phosphorylation. So that's why it's not possible to reach the plateau to the negative.
Perfect. Thank you so much, Elodie. So it appears as though we're at the end of the time allotted for questions. So thanks to everybody who submitted questions. And thanks so much to everybody for attending, and again for Elodie for presenting details of her research today. So thanks again, and have a great day, everybody. Bye-bye.